Right, as president, it gives me great pleasure to present you with this certificate for the Hammond Award, which is for the best article in the AES Bulletin on a British species. Congratulations. We can do it again.
Okay.
Smile at the. So you'll be in the bulletin.
Thank you so kindly.
Thank you. Well done.
No, this is
more important. This that Charlie is former bug clubber.
Ah, right.
yes. Very proud former bug clubber and still a bug clubber in spirit, I add. I'm very grateful for this, and it couldn't have actually come at a better time because I'm aware Hammond illustrated a beautiful key to British Odonota to dragonflies quite a few many years ago, and I was looking to purchase it. So the prize from this reward, that'll be a fitting direction for that. But anyhow, I'd like to give this presentation and thank you all for your time. The subject is a ground beetle sampling project for redback shrike reintroduction in Sussex. So UK biodiversity in general has significantly declined since the 1970s. And a lot of people assume that insects are the most affected taxonomic group. And whilst this is true for population numbers, birds are actually classed as the most threatened biodiverse group, 43.3% of 293 species having declined since the 1960s, and many species of British bird completely vanishing in the last decade. And one example of this is the red-backed shrike, Lanis colurio, which is a migratory bird famous for its unique feeding habits. It's earned the vernacular the butcher bird because it picks up prey items and impales them on suitable twigs, storing them for later. One important group of victims have been identified as beetles in the family Carabidae, exceeding the length of 9 millimeters, which make up a huge group of the food base for these birds, specifically beetles in the genus Pterosticus, Poecilus, Carabus, and Harpalus. Fortunately for beetles and mice, this bird is actually considered functionally extinct in the UK, as they've stopped breeding since the 1990s. This is because of habitat loss, decline in insect populations, and the pastime of egg collecting, which obviously detracts from the population when it's most vulnerable. So in the UK, the last time these butcher birds were recorded as a significant breeding species was the 1970s. However, it's 2026 now, and the Knepp Wildland Foundation would like to reintroduce the redback shrike to its former ranges in West Sussex. And of course, one of the key elements to any successful reintroduction program is the availability of a suitable habitat and an adequate prey resource. And there was a bit of a research gap before this project as to where this suitable habitat would be and what the adequate prey would present. So 4 study sites were selected because of their scrub habitat, which is the traditional space of living for the redback shrike, and the air abundance of carabid beetles. The sites are as follows: the honey pools, which are a tract of scrubland habitat in the Knepp Wildlands; Fair Oak Farm, which is a smaller rewilding project in Limkin; the Iford Estate in Houndine Farm, which is another former intense agriculture turned into a rewilding project; and Pepper's Combe Nature Reserve near Staining. These sites represent a range of ideal conditions, management histories, and landscape contexts across Sussex. They're also vital geographic stepping stones needed to make the 100-mile Weld to Waves corridor, which is a habitat project, a charity which intends to produce a continuous and functional 100-mile nature recovery corridor of scrub across Sussex. And this is a really exciting project, but it relies upon successful collaboration with local organizations like Weld to Waves, Sussex Wildlands, to deliver practical projects. And it's very grassroots, involving lots of volunteers. I was not involved in the sampling of the beetles. It was conducted by volunteers from Well to Waves and students from Sussex University. However, I was well informed of the sampling methodologies as they were crucial in understanding the insects which I later prepared in the museum. They were as follows. The preferred sampling areas were mosaic grassland with nearby large scrub for the birds to nest in the edges of, numerous perches, dead branches, and bare ground, which is where they conduct their hunting. Other insect groups were collected in this project. However, in the interest of this talk, the beetle sampling methodology will be described. 5 trapping locations were set up and pitfall traps were dug. This came to a total of 25 pitfall traps and 15 pan traps for Hymenoptera per site. However, of course, for the purpose of this talk, the Coleoptera traps will be discussed in greater detail. They were designed as followed. You can see in the figure with a corrugated iron mesh on top to prevent larger animals like amphibians, reptiles, and rodents from falling inside, a nifty rain cover, and 4 stokes to secure the cover, right? They were part filled with water and washing up liquid to trap any insects which fell in. And they were raised slightly, you can also see, above the level of the ground, so that when insects crossed over the precipice of the lip, they fell directly in. Then the material, after a week of overnight trapping, was selected and sorted through on site. It was emptied into a cup or a bowl, and detritus and any bycatch not of interest, so specifically slugs and worms, were sorted out, and all the other invertebrates were retained and emptied into individual pots with a premixed 70% ethanol solution. The worst invertebrates to find in your pitfall traps are slugs, which become scarily bloated and spread slime all over the other specimens. Each trapping location was, of course, labeled and kept separate and clearly indicated with the paper label inside the tube and written on side. I came in at around this point because the beetles were stored in the Sussex Uni Living Lab and they didn't want to hang on to the specimens anymore due to the limited storage space. The total sampling actually produced 4,750 carabid specimens in 315 pots, which were identified to species level. On top of this, there were another 40 pots of unprocessed beetle specimens, which hadn't been sampled. So Rachel Bicker, who's an ecological consultant and rewilding ecologist, kindly donated them to the Natural History Museum Coleoptera collection. And I was instructed to set aside a minimum of 60 beetles each, 30 male, 30 female, from each of the 4 sites, applying to the following species, which are illustrated on the left here. Abax parallelepipedus, Pterosticus medidus, Pterosticus niger, Pterosticus melanarius, Harpalus rufipes, Calathus fossipes, Postulus cupreus, and Postulus versicolor. This was a lot to prepare, and I had difficulty producing mounted specimens, which are both cosmetic, which we all know can take a lot of time, whilst also efficiently processing all this material. The majority of beetles were mounted on sea-white card, glued to a board with gum tragacanth, and pinned with size 2 to 3 Austerlitz insect pins on plastizote unit trays. When set, they were then transferred into wooden display boxes in the collection. The majority of identification was conducted using the key, the Carabidae of Britain and Ireland, by Martin L. Luff. And with the help of this, we were able to confer this sort of beetle soup into well-presented labeled specimens. And you can actually see in the top plastizote tray on the top right, there were quite a lot of non-target species, so carrion beetles and elaterid beetles, which I'll cover here. When we look at the beetles observed, I've encountered all of the required ones except for Calathus fossipes. So far, I've also processed only 20 pots of the 400. So it's probably a matter of probability that I have not bumped into them yet. The samples were dominated by carabids, particularly the species in the genus Pterosticus and Postulus. Pterosticus medidus, Pterosticus melanarius, and Pterosticus niger were all well represented. However, these are generically common species of ground beetle. I'm sure a lot of you anecdotally recognize them when lifting over logs in the forest. They're relatively large and common predatory ground beetles associated with grassland, scrub, and field margins, but would also constitute substantially good prey items for the redback shrike. Postulus cupreus and Postulus versicolor are really beautiful in my opinion. They were also very frequent. And I have a prepared tub of them here I'd like to pass around. The light is a bit dim here, but if you shine your phone torch on, you'll see the remarkable iridescent colors on the pronotum and carapace. On top of that, several interesting beetles were observed that weren't part of the target assemblage. Of course, Carabus violaceus, which you can see the genus name is blocked by the convenient talking NHM zoom indicator. But yeah, Carabus violaceus, which is the biggest group of the beetles, recorded at 20 to 30 millimeters, and also some carrion beetles like Silpha carinata and Phosphuga atrata, and some interesting elaterids, many of which I have not been able to identify because that group is quite difficult and quite time consuming, and frankly I was unable to process it in the amount of hours given. But for instance, Scopaeus species here, unidentified, which is particularly impressive to look at, very structurally interesting. So what did we learn from looking at the beetles? Overall, Houndine Farm is supported by the greatest abundance and total biomass of carabid prey, with beetles accounting for more than 80% of the recorded biomass in total when they sampled there, and a lot of medium to large-bodied carabid beetles, which would be substantial protein snacks for the shrike. NEP supported fewer total insects. However, it covered a greater proportion of larger-bodied prey items. In essence, the beetles were bigger. As a result of this, we have to make the conclusion that no single site is completely optimal for reintroducing shrikes. Each has its own benefits and trade-offs, which are listed below. Whilst one site, NEP, was less productive in terms of numbers, it contains a higher proportion of larger scrub-associated ground beetles, which may support the adult shrikes. However, in trending towards that kind of species assemblage, what will the shrikes feed their chicks? They may need smaller individuals of insect, in which case houndine might provide better. In total, ideally you'd introduce on several sites and then compare progress, or perhaps relocate birds that aren't doing so well on one particular zone to another. But as aforementioned, reintroduction involves a lot of legislation, a lot of red tape, and multi-site reintroduction could be costly and extensive. At the moment, the Net Wildlands haven't actually publicized which site they'd be interested in reintroducing, and most likely further research or biological recording will be necessary to make a solid conclusion. Of course, we can't acknowledge this conclusion without making case of the limitations of the study. One important feature is that pitfall traps capture different invertebrates unevenly, so they represent a hidden bias. For instance, faster and larger beetles, such as those in the genus Carabus, are more likely to be caught in a pitfall trap than smaller, less active beetles like elaterids or clicks. And this creates a bias in the sample where certain groups will appear more majoritatively, not because they're more abundant in the scrubland, but just because they fell into the trap more. Second of all, pitfall traps don't actually represent shrike hunting patterns very well. They don't give us such a good indication on if the shrikes will be able to access and enjoy this beetle prey base. To explain, shrikes don't dig a hole in the ground and wait overnight for their food to fall inside. They're diurnal predators, and a lot of the beetles recorded will be nocturnal, as they're traveling about in the scrub at night and falling in. As a result of this, it's uncertain as to how many of the collected carabids the shrike would actually be predating on. I predict Postulus, as these are also a diurnal species. You can often see them, especially in parks and public recreation spaces, running around in the sun. And finally, of course, invertebrates smaller than 9 millimeters were discarded. And from my perspective, this represents a loss of valuable specimens because there's a lot of interesting material from the taxonomic point of view that was totally filtered out and discarded. As you can tell, there was quite a repetitive, large group of macroinvertebrates collected and not a lot of smaller microinvertebrates, interesting beetles like that. Another thing that's important to note is that beetles smaller than 9 millimeters are caught and provided by the adult shrikes to their nestlings. So in actuality, they depend not only on the large-bodied carabid prey base, but on the smaller specimens, which we have no clue as to how abundant they are. And that concludes the talk. I hope you found it informative, and I thank you for your time. I'd like to acknowledge thanks to Rachel Bicker, who donated the specimens, Walter Waves, and the Net Wildland Foundation, who funded a lot of this research, and Sussex University, whose students provided their valuable time and study hours to assemble the beetles. I'd like to thank the Natural History Museum and their Coleoptera team for guidance in identifying and preparation, and the AES, both for this wonderful award and for hosting me about this talk. I'd also make a totally selfish and unrelated side note. I have a tub of Atticus atlas caterpillars that are in search of a new home. And they're currently residing on the Queckit Microscope Club table outside. If anyone would be interested in rearing these beautiful moths, they feed on privet. They're first instar at the moment, so very small, but they will get bigger. They're about as simple as most of the very easy phasmid species like Periphasma sheltai, and they produce the most amazing big kind of decorative moth species, which you can either sleeve, you can put them in a cage, or you can just stick them on a house plant. And they're pretty immobile. They don't move around a lot. So they make quite a interesting conversation piece at any dinner party. Thanks again.
For Charlie. How do they how do the strike get introduced, and do they return to the same site following year if they're summer visitors?
Presumably so. They'd be reintroduced from captive bred European populations, which are usually kept in, I think, zoological institutes, like in Vienna or places like this. They have captive populations in aviaries that could be reintroduced. And rather much like salmon reintroduction programs, there is a lot of skepticism as to when you have a captive migratory species, whether it'll migrate when assembled into the wild. But usually they actually realign.
Absolutely. Yeah, thank you very much. I really enjoyed the talk. It was really good. So I just wanted to ask you about that slide about the different sites and saying that no single site seems to be optimal.
Yeah.
So there was one site, Hammond where you had greater quantity of prey and net basically larger prey.
Yeah.
I just wondered if you had any thoughts about why there's that difference and whether it was to do with vegetation succession or something like that. Is it management related? Is it?
I think it could be management related. I think, on the Hammond site, you saw a greater sort of, there's a lot more saproxylic beetles,
right.
A lot more dead wood, and that's generally going to support a greater biomass or abundance. Whilst on NEP, you saw, especially the sites that were recorded, you saw less dead wood coverage ground, and that could have been one reason why, because generally speaking, where you have less dead wood coverage, the larger individuals are going to be less vulnerable. I correct myself. The larger individuals are going to be more vulnerable. They're going to have less places to hide. So, obviously, NEP. As I say, I need to actually reciprocate that point. NEP had great dead wood coverage on the other side,
right? Okay.
So larger individuals were less conspicuous, probably be less vulnerable to avian predators. Was Houndy, you had a lot of grass, a lot of scrub, but perhaps smaller individual beetles and calibers would be hiding the larger ones will still be conspicuous, will be picked off, and their population is smaller. But also, I mean, it could be to do with the time of year, for example, things recorded.
Were they single tracks each side? Or
They were, I believe, just to clarify, yeah, they were. I can give you the direct number. So it was 5 traps a site, and they were as equidistant as possible. See here, this was in the Fair Oaks Farm trapping station. So obviously, in actuality, it's impossible to situate the traps so they're perfectly apart. Nonetheless, as good as possible. Also recognizing the Hammond there. So there's 25 traps in total. That's all the sites. 5 sites.
The differences between traps of the same size, relatively small, sort of relatively consistent. As in like at one site, did all the traps say, were the differences between traps smaller than the differences between sites on average?
The differences between individual traps.
Yeah.
Yes, actually, it was. So anecdotally, I can say on a lot of sites, you find one trap had been largely composed of a particular taxonomic group, right? So a particular genus or like a particular family of beetles, right? And then on another trap, you find a totally different genus. So one trap would be dominated by Posilus, the metallic ones that were passed around. So you can see that trade came entirely from a single tube. So there was a tube from one trap that was dominated by a single genus of beetle, effectively, with very few Pterosticus inside. And then on the other hand, you find traps that were largely dominated by Pterosticus, bigger bodied ground beetles, probably due to proximity to dead wood. I'd imagine some traps are situated closer to a log or something, and you find more bigger bodied beetles, whereas the traps that are situated in the middle of grassland, equidistant, less so. I do believe they tried to minimize this variation, but of course they couldn't clear habitat with the stone. So at the point you have to acknowledge the just heterogeneous quality of nature and trapping in the wild. There will be a degree of uncertainty, lots of different fluctuations, right? Habitat type you can't sometimes accommodate that realizing to the trap.